Hello everyone. In today's module we will discuss about fermented milk. The primary objective of this module is to make you familiar with the microbiology and biochemistry of fermented milk. As we know that the demand for fermented milk product is always rising due to their reported health benefits and nutritive values. The major prerequisite for fermented milk products include that they should have low caloric values, palatable, refreshing, smooth in texture and with extended shelf life. In this module we will discuss an overview of milk as balanced nutrient diet with an emphasis of technologies to enhance the nutritive value of raw milk to confer potential health benefits.
The fermentation of milk enriches it with protein, other essential amino acids, essential fatty acids and vitamins. Additionally, the fermentation eliminates anti-nutrients component in the raw milk. Fermented milk products have enhanced flavor and aroma with enhanced digestibility and health promoting benefits. Fermentation of milk may assist in the destruction or detoxification of certain anti-nutritional compounds, if present. The health benefits of fermented milk include reduction in cholesterol, anti-cancer effects, immunomodulatory effects, longevity, anti-hypertensive effect, anti-diabetic effect. Therefore, this module throws light on different milk processing technologies and gives a way for further scientific and research analysis.
Milk is a rich source of nutrients and biologically active compounds which provides the immunological protection to the infants by improving the digestive maturity of the infant's gut, development of gut-associated lymphoid tissues and symbiotic microflora.
The milk is comprised of proteins, lipids, lactose as major sugar, minerals and vitamins. However, the composition of milk is affected by species breed, feeding of the breed and stage of lactation. Milk also contains important fatty acid, namely linoleic acid, linolenic acid that are synthesized during biohydrogenation of fatty acids present in the animal feed. It is also a richer source of casein and other whey protein, vitamins and mineral components such as calcium, copper, iron, manganese, magnesium, phosphorus, sodium and zinc.
Lactic acid bacteria represent a major group of microbial community which has been the most extensively studied microorganisms in milk and fermented milk products. Most of the lactic acid bacteria present as autochthonous microflora of the milk, or these can be added as an inoculum in fermented milk.
The milk fermentation process is reported to be relied on the activity of lactic acid bacteria, as this group of bacteria play a crucial role in converting milk as raw material to fermented milk products. The milk fermentation industry relies on the characteristics of various strains of the lactic acid bacteria used as starter cultures. For an initial inoculum, the starters are obtained from sequential steps with respect to isolation of cultures, followed by activity rating test and followed by its growth in milk and quality of the finished product. The most important properties of lactic acid bacteria is the acidity rate and production of metabolic products such as flavoring compounds. Lactic acid bacteria starters, due to their metabolic activities in milk, provide taste and texture of fermented milk products, inhibit food spoilage bacteria by producing growth inhibiting substances such as organic acid, bacteriocins, etc. A wide variety of fermentative lactic acid bacteria are involved in the production of fermented milk products such as yogurt, cheese, cultured butter and sour cream.
Since milk is considered as highly perishable commodity, therefore in older times fermentation of milk was carried out to preserve or extend the shelf life of perishable milk without the proven facts or health benefits of fermentation. During those days, An inoculum of older fermentation was used to prepare fresh batch of fermented milk product, which has been replaced by application of direct wet starter DVS cultures. Apart from extending the shelf life of the milk, the fermentation of milk also results in enhancement of organoleptic qualities of the milk, functional properties of fermented food product, nutritional and economic benefits.
Lactic acid bacteria as starters. Starters are defined as cultures which are used in fermentation for desired changes in raw substrate. Since the milk is usually fermented with the help of lactic acid bacteria, this module focuses on different lactic acid starters, their metabolic activities and application in the production of fermented milk. Lactic acid bacteria group encompasses various species of lactobacillus are used as starters. Other lactic acid bacteria also include Leuconostoc, Pedococcus, Lactococcus, Streptococcus, few species of Carnobacterium, Enterococcus, Onychococcus, Teredo, Genococcus, Vagococcus and Vacella.
Acid production. Production of organic acid as an end product is possessed by all the lactic acid bacteria by carbohydrate metabolism. Based on the end products, the lactic acid bacteria are divided into two categories: homofermentative and heterofermentative. Homofermentative species of lactic acid bacteria converts milk sugar in milk into lactic acid as primary end product, whereas the heterofermentative species convert milk sugar into lactic acid, acetic acid, ethanol and carbon dioxide.
Both Rhodes and Cocci follow different pathways of carbohydrate fermentation and achieve end product which are at least 90 or 50% lactate in case of homo and heterofermentative species respectively. The homofermentative group includes various strains of Lactococcus species which results in rapid development of lactic acid. Other homofermentative lactic acid bacteria include Lactobacillus delbrueckii subspecies bulgaricus, Streptococcus thermophilus, Lactobacillus helveticus and other Streptococcus species, Enterococcus, Pedococcus and Aerococcus. In general, the heterofermentative lactic acid bacteria are rarely used as dairy starter cultures, as at significant numbers these might result in defect in milk and milk product. This group includes Leuconostoc species, Lactobacillus brevis, Lactobacillus fermentum and Lactobacillus rotari. Other Lactobacillus species are considered facultative heterofermentative, as these species produces carbon dioxide and other byproducts such as Lactobacillus blantarum, Lactobacillus casei and Lactobacillus curvatus. Other metabolic pathways. Few lactic acid bacteria are reported to ferment other substrates such as citrate, gluconate and certain amino acids. The fermentation of citrate resulted in the production of flavor compound such as diacetyl, Leuconostoc mesenteroides subspecies cremoris and Lactococcus lactis subspecies lactis biovar diacetyl lactis. These species are often used to make products such as buttermilk, sour cream and cultured butter. Hexoses other than glucose such as mannose, galactose and fructose are utilized after different isomerization and phosphorylation steps. Galactose is utilized via conversion to galactose 6-phosphate through phosphoenol pyruvate dependent phosphotransferase system, tactose 6-phosphate pathway, or as free galactose through permeases. Production of flavor compounds. Lactic acid bacteria possess an array of enzymes with hydrolyzing capacities towards all the nutrients such as carbohydrate, proteins, lipids into different aroma compounds. Carbohydrates are fermented via homo or heterofermentation depending on the lactic acid bacteria species used in the fermentation. This fermentation leads to the production of lactic acid as an end product and other several aroma compound or aroma precursors such as acetaldehyde, acetic acid and ethanol. Citrate An important component in milk is metabolized by citrate utilizing lactic acid bacteria, resulting in excess of pyruvate. The pyruvate therefore produced is converted to diacetyl acetone and 2,3-butanediol. In this slide, you can see metabolic pathways for production of flavor compounds by lactic acid bacteria in fermented milk.
Proteolytic activities. Lactic acid bacteria are also able to synthesize amino acids at low levels. Therefore, these are fastidious organisms where they are dependent on the other sources of amino acids. The proteolytic activity in dairy starters is well recognized. The lactococcal proteolytic system consists of membrane-bound enzymes, transport system, and intracellular peptidases cleaving the proteins to smaller fragments. The proteolytic activity contributes to the development of the flavor, aroma, and texture of fermented products. Various types of peptidase such as endopeptidase, dipeptidase, aminopeptidase, tripeptidase, and proline specific peptidase have been reported in different strains of lactic acid bacteria.
Categories of fermented milk. The general steps in the production of fermented milk include selection and standardization of milk base with low total solids, selection of additives like fruit juice, concentrate, colors, artificial sweetener, sugar, stabilizers, heat treatment of the milk, selection of appropriate starter mix with high acidification rate, low ropiness, and gas production. Once the starter is added to the standardized milk, the fermentation results in acidification down to pH 4.6 for a complete casein coagulation and product formation. Here you can see different fermented milk with their starters. Acidophilus milk. Acidophilus milk is a low-fat or non-fat milk fermented by Lactobacillus acidophilus. The acidophilus milk is considered as a dietary adjuvant which is reported to control of intestinal infections, maintenance the microecology of the gut, lowering down the cholesterol levels, reduced risk of cancer, and enhancement of bioavailability of key nutrients. During processing, the milk is heated to high temperature and inoculated with Lactobacillus acidophilus at the level of 2 to 5%, incubated at 37 degrees Celsius until an acidity of 0.6 to 0.7% is achieved. As the acidophilus milk is not good in the aroma, a variation has been introduced as the production of sweet acidophilus milk where no incubation is done after addition of starter. The milk exerts its effect when the starter reaches the gastrointestinal tract, delivering different health benefits.
Traditionally, the cultured buttermilk was prepared by addition of lactic starter in the byproduct of butter manufacture, but the modern technologies uses skim or whole milk for the production of this product. Usually, the milk is fermented using Lactococcus lactis or Lactococcus cremoris. The milk is primarily heated at 95 degrees Celsius and cooled to 20 to 25 degrees Celsius before the addition of the lactic starter at the rate 1 to 2%. The mixture is incubated 16 to 20 hours up to the acidity of 0.9% lactic acid.
Bifidus milk. Bifidus milk is reported to be nutritionally superior with improved digestibility and palatability. Among the genus Bifidobacterium, the commonly used species in the production of Bifidus milk and Bifidobacterium bifidum, Bifidobacterium infantis, Bifidobacterium longum, Bifidobacterium adolescentis, and Bifidobacterium animalis. Bifidus milk can be prepared from skimmed milk, partially skimmed milk, or whole milk. The final set product has a pH of 4.37 to 4.7 and contains viable cells of Bifidobacteria. The Bifidus milk can be produced as a stirred product.
Kefir is a fermented dairy product with a combination of lactic acid and alcohol fermenting bacteria. This product originated from Caucasus region. Its grains are rich in nutrients including folic acid and potential health benefits. The fermentation is a basically spontaneous fermentation resulting from the introduction of kefir grains into raw milk. The kefir grains are small, spongy, symbiotic colonies of yeast and bacteria.
Komis is a fermented product made from mare's milk. Komis is native to parts of southern Russia. The milk is fermented with a liquid starter culture that may comprise of Lactobacillus, Lactococcus, Streptococcus, Leuconostoc and Pedococcus. For Komis production, the milk is heated at 90 to 92 degrees Celsius for 5 minutes and cooled to 26 to 28 degrees Celsius. After cooling, the starter is added at the rate of 1 to 2% incubated and cooled to 20 degrees Celsius while stirring for 1 to 2 hours. The fermented product is bottled and allowed to ripen at 6 to 8 degrees Celsius for 1 to 3 days. Based on the different concentration of lactic acid and alcohol, 3 types of komis can be prepared as weak komis that contains 0 to 7% lactic acid and 1% ethyl alcohol, ordinary komis 1.1% lactic acid and 1.8% alcohol, and strong komis 1.8% lactic acid and 2.5% alcohol.
The importance of fermented dairy products in modern day life is underlined by the wide varieties of milk products marketed globally, not only for the benefits of preservation and safety but also for higher economic growth along with higher sensory attributes. The microflora responsible for the fermentation of milk is indigenous and comprised of mainly lactic acid bacteria. Besides, information about the implication of the presence of their metabolites and their chemical interaction with milk components is still insufficient. Currently, the milk fermentation procedures have been used to develop the traditional practices involved at the commercial scale. However, more efforts and research is required to develop novel fermented milk product with enhanced health properties.